Great. Absolutely. And thank you, Dr. Lai, for the opportunity. We're always striving to enlarge the practice with the pain management in the department, which is a very good opportunity so we can provide a better care for the patients here in South Florida. I mean, it's a bit of a challenge to introduce Ricardo Vallejo because his extensive CV, but I could start saying that he did his MD, PhD in Madrid, Spain, and then he came to Miami where he did his residency at the University of Miami in anesthesiology. So I think he's coming back home. After that, he went to Harvard to do his fellowship in pain management at the Mass General. And after that, he went to Illinois in Bloomington as an adjunct professor at the Illinois State University, where he did a very extensive research related to immunology and pain. After that, he became the director for the research at Millennium Pain Center, where he ran over 150 clinical trials, which is amazing. I mean, if you look at the papers he published, which are over 160 papers, he's an icon in the management of pain, especially with neuromodulation. With the DTM coming up, he'll explain this coming later. Over 20 book chapters, 2 books himself, and now he became the CEO for 4 companies, and now I guess he's coming back to Florida, where he lives up north here, like half an hour from the medical campus. And Ricardo, please, it will be a pleasure to hear you talk about neuromodulation.
Thank you very much, Bashir. Thank you, Alan. I have to say, I also have to thank Dr. Lardi. I typically always like to do presentations, virtual presentations is easier. But at the same time, this particular presentation today has an emotional component to me, and is to be back in Miami and be talking with residents and fellows, attendings from University of Miami. That's, again, very emotional. Unfortunately, I'm not in campus. That was my first suggestion. Before I start, let me give you my first slide with a conflict of interest. Joashir mentioned something about my entrepreneurial hidden part of personality that I never used until the last 7 years or so, when some of my patents developed in basic science ended up being translated into clinical results that I'm going to show you today. Today, I'm going to try to be as clean as possible regarding my research. I think what is interesting for me and what is worth for me to discuss is what do we do when we stimulate the central nervous system at any location. What are we really doing? We are used to think about parameters from an electronic point of view. I apply an electrical signal, and then that leads somehow to depolarization of neurons. I'm a Spaniard, and maybe because of that, I'm very sensitive to the neuronal doctrine that dominated almost from a century everything related with our field, and it had to be with to do with Ramon y Cajal. And when you think about it, it basically centered all the communication from the outer world into our brain through these neurons that communicated each other. That kind of presented the central nervous system as just dominated by the neurons. That's how we are talking today about neuroscience, neurology, neurosurgery, neuromodulation. Everything has to do with these rock stars in the field. But let me tell you a little bit briefly in terms of the neuromodulation. How did we came where we are, and what are we trying to do right now to change this paradigm in the field? This on the left-hand side, and I think you can see my mouse, you see the original diagram or cartoon image that Patrick Wald made to Ronald Melzack back in 1965 to describe how the gate control theory would work. For those of you that may not be very familiar with the concept, it's relatively simple, and this simplicity is what led to the to every physician in the field to take those concepts and bring it into the neuromodulation. You just had to stimulate A beta fibers, therefore producing paresthesias. And by doing that, you will close the gate at the level of the dorsal horn for afferent nociceptive information transferred from small fibers, A, delta, and C fibers, to bring the nociception. Very simple concept, and that's why it was so easily embraced. Now, if you think about what the only thing that you need to do is you need to apply an electrical current that is high enough, intense enough, and that has certain duration that will generate an action potential. As long as you do that, you will generate this action potential. If it happens that the duration is shorter or the intensity is not long enough, you will change the resting membrane potential. But if it doesn't get to the depolarization threshold, then no action potential will be generated. Very simple, and that's why people love it and embrace that into the practice. This is a very famous landmark study by Richard North back in 1991. What he observed in this study, this clinical study, is that there was a significant association between the generation of paresthesias in the painful area and the alleviation of pain. That seems to be the big conclusion of the study. But if you look in detail, you will see that there will be patients with 100% coverage with paresthesias of the painful areas who had no relief whatsoever. And you will also get patients with only partial coverage of the paresthesias with the painful areas. And despite that, they have excellent relief. In reality, if you look at the correlation coefficient, it was only 0.35. So not strong at all, but once again, under the assumption that the paresthesias were necessary, that was interpreted as a conclusion of the manuscript that you needed to get this paresthesia matching the areas of pain. If you think about it, it's like a marching band. You stimulate, and then you create a depolarization, you stimulate, you create a depolarization, and kind of all the neurons synchronize in generating those messages up into the brain, and therefore alleviating the pain. One of the challenges, though, was to match the paresthesias with the pain. Those are early studies in which one of the challenges that they observe is that in order to stimulate the lumbar region, which was one of the most common patient symptom presentation, they had to stimulate fibers in the L2 to L4 area. But when they tried to stimulate, for example, at L1, the fibers were still too lateral, which means that fibers that were coming from other regions were more easily stimulated, or if they put the electrical signals too lateral, they will be close to the dorsal root ganglia, and therefore they will producing uncomfortable paresthesias at the amplitude necessary to cover the lumbar region. That made that slowly people started to put the leads higher and higher up, and it was considered that areas around T9, T10 were the best ones to generate the paresthesias, because those fibers from the L2 to L4 will be more midline and therefore easy to stimulate without the side effect of the stimulation of the dorsal root ganglia. Another interesting observation is that when they were trying to do that, they also observed that the fibers that were nociceptive in nature, those fibers with a small diameter, like 7 microns or so, were extremely little in terms of the amount of these cells in those regions. They had a lot of fibers of larger caliber, but not the fibers that they were trying to stimulate based on the gate control theory that formulated that you had to stimulate in the dorsal columns these intermediary neurons of a small caliber. So there were a lot of challenges and a lot of advances trying to optimize this coverage with paresthesias of the painful areas. This is the strength duration curve. I just presented for a simple reason. There is two old concepts that we don't use them anymore longer, but one is Riobase and the other one is Chronaxi. The strength duration curve basically tells you that in order to avoid the stimulation of large diameter fibers, which are more easily stimulated at lower amplitudes, you have to increase the pulse width, because as you can see here, the Riobase is the minimal amplitude that will be required to produce a depolarization of the neuron. If you go towards the right side where pulse widths so the duration of the electrical impulses are larger, the separation between small sensory fibers and motor fibers are much closer, so it will be easier here to obtain depolarization of these sensory neurons versus the opposite, trying to stimulate with low pulse widths, which will require very high amplitudes. All this was good, but I have to say that since I started in the field and I got very inspired by the gate control theory, it was clear that the main purpose of the parameters that were formulated mainly through computational models or finite mathematical models were to optimize the parameters to lead the depolarization of the neurons. And if you obtain the depolarization of the neurons, then you will cross your fingers and you will hope that the patient will get alleviation of the pain. That's quite interesting, because if you think about any other condition, the fundamental aspect is to understand what is the pathology that you are treating. Once you understand the pathology, then you can formulate different types of therapies. But here we are trying to understand a theory. Based on the theory, we formulate a therapeutic approach, and then finally, we come to the conclusion that that should be helping our patients. This is a study that I participated in. It was published back in 2014. And the reason I bring in the article is because they compared something called high-frequency stimulation at 10 kilohertz versus the use of conventional stimulation, therefore generating paresthesias in the painful areas. Beyond the significant improvement or statistically significant better results with the use of high-frequency stimulation at 10 kilohertz versus 50 hertz, what is very interesting is that these patients did not have paresthesia. And please keep that in mind as we move forward. And the reason is because the amplitude that was used to treat those patients was extremely low. They tried high amplitudes trying to generate paresthesias, but the sensations were very uncomfortable and patients could not tolerate paresthesias at these high frequencies. This is some search for a mechanism of action. Now we have a problem. There is no longer paresthesia, so the gate control theory doesn't fit any longer. So what the company has been trying to do for the last 8 years or so is try to figure out how does it work. This is an animal model where they stimulate the hind paw of the animal. They have a recording electrode at the dorsal horn, and they have an electrical stimulation over the dorsal column. And as you can see, as you apply different types of stimulation, you will get different recordings from these neurons that are underneath the electrode. And if you have like noxious or pinch stimulation, you will have the activation of these all these ectopic action potentials. What they observed is that there were two different types of neurons that they could be stimulating. One that were not adaptive or inhibitory neurons, and others that were excitatory neurons. And they also observed something quite interesting. If they use the same 10 kilohertz, at lower amplitudes, they were able to stimulate or generate activation of the inhibitory neurons, while the use of higher amplitudes ended up stimulating excitatory neurons, something that in theory, at least, you don't want to generate. So they kept working on this, and they compared with different frequencies, CHAM or 1000 hertz or 10 kilohertz. And they always observed that in the dorsal horn, keep in mind that the stimulation is in the dorsal column, The dorsal column and the dorsal horn in the rat are very, very close together, but they seem to observe that they had more reduction of the wind-up phenomenon or the all these ectopic action potentials when they were using low amplitudes of high-frequency stimulation. And they kept studying the same concept and basically concluded that the only way that it could be working, this therapy in the clinical world, is that at low amplitudes, they were activating these non-adaptive inhibitory neurons. So that's kind of an explanation for that, except that in humans, the distance between the dorsal horn and the dorsal column is much larger. But again, it created something quite interesting from my point of view, because at that point, since 2004, my lab was focused into the molecular mechanisms subjacent to pain and how electrical signals could affect that. Very briefly, I'm going to present another theory that has been formulated, which is the activation of a specific supratentorial pathways, in this case, medial pathways, by a form of stimulation called the burst stimulation versus other forms of stimulation. This is quite interesting from a literary point of view, because in a study with 15 patients, 5 patients were selected. And after about 8 years, the same 5 patients were presented isolated from the other 15 in a study showing these EEGs and activation of the medial pathway when using this type of stimulation. Unfortunately, nature doesn't work that way. And if I always put an example, if I throw a stone in anybody's toes, you're going to have an immediate activation of the medial pathway. So I don't know if you really want to get this type of effect or not, but that was formulated as a mechanism of action of another type of therapy. At the end of the day, science always tries to move and provide different types of responses. So this is a study done with PET scans. And what they did is they did 10 patients. They look at whether or not there was activation of different areas of the brain with the use of spinal cord stimulation. And interestingly, with paresthesia-based stimulation, they also observe activation of the same region. And since then, there are 2 studies, one done by Dr. Resai and the other one by the Groth. And the 2 of them observed, once again, with paresthesia, activation of the medial pathway. In other words, it seems like we have been searching for a mechanism of action to try to somehow improve the way we can provide a therapy. But the search for that mechanism of action has been a little bit complex, to say the least. This is one of my favorite quotes, and this New York writer once wrote that you can ignore reality, but we cannot ignore the consequences of ignoring reality. And I just want to make a little pause here to describe a summary of what I have been trying to tell you. We came with a theory, then the theory is somehow disproved by these non-paresthesia-based formulations, whether it's 10 kilohertz or the so-called burst, which is at 500 hertz, both of them not generating paresthesia, but with not a very clear understanding of what's going on. And when I talk about the ignoring reality, is because something has happened over the last 5 decades. My background is in immunology, so maybe that's why when I got into pain medicine, or I started looking into medicine, I immediately start thinking about pain as an inflammatory process, and therefore there has to be some inflammatory component that is modulated. This is what happens. This is a very old cartoon from 2009. I keep it because it's simple. Compared with the reality is much more complex. Basically, if you have a continuous nociceptive injury or information coming into the dorsal horn, the presynaptic neuron initially is going to start releasing some excitatory neurotransmitters, and those neurotransmitters will be recaptured by the same type of receptors in the membrane of surrounding cells, which are the glial cells, and they will clean the synaptic cleft and maintain the hemostasis. If the injury continues or those nociceptive information keep coming to the synapse, then you're going to start activating first the microglial cells. You will generate epigenetic changes with the generation of different kind of pro-inflammatory cytokines that will activate the astrocytes. And when that happens, these epigenetic changes leads not only to the downregulation of those receptors in the membrane of these cells, but also to the generation of pro-inflammatory cytokines through different intracellular cascades. This is what happened when patients develop chronic neuropathic pain. But as you had seen, I'm almost at the middle of my presentation, I had not talked about the condition that we have been treating. I've been talking about how do we optimize neuronal depolarization, or what is happening when I apply these so specific parameters based on the gate control theory, but we have not discussed at all what happened when you have pain. And I think, like in any other condition, that's where we should have started this process. This is a paper from 1994. What they observed is that if they injured the sciatic nerve ipsilaterally, they will observe that there will be an activation of a marker of astrocyte activation, which is the GFAP. And they did the injury and they observed ipsilateral activation of these cells. Interestingly, when they inhibited the NMDA receptor, which is one of the receptors involved in the wind-up phenomenon, they did not only decrease the pain of the animals, but there was a correlation with a reduction in the activation of these cells in the dorsal horn. When you use in vitro a set of glial cells, and you apply different parameters, you will observe different effects. Here they are looking for the release of glutamate from astrocytes. And as you can see, different frequencies, different current intensities will generate different amounts of glutamate released by those cells in vitro. Same way, if you are just looking at the action potentials, you will see that there is release of glutamate when you apply electrical signals. And if you apply an electrical signal to block this release of the action potentials and also the release of glutamate, you will see that here, when you apply the electrical signal, there is a still release of the glutamate. But what happens if you use, for example, a sodium channel blocker to only block the neurons? What you will observe then is there is no longer ectopic action potential. Your electric recordings will not be able to record those any longer. But interestingly, glutamate is still released, which means that there is something beyond the neuron that is causing the release of this main excitatory amino acid. I love this slide. This is from the 1990s. And it's because going back into the neuronal doctrine, you will observe that initially, if you think about a neuron, you will think about this cable that is communicating with the other. Somehow the electrical signal converts into a chemical signal that is released in this extremely minute space at the synaptic cleft. These chemical signals will be converted into action potentials, and that's what happens. This is kind of the definition of what a neuron does when it's transmitting information. But here you see this is a plate that is full of astrocytes. There is only astrocytes, and then they add glutamate. And when they add glutamate, initially you see activation just where you apply that. Interestingly, if you think about, because that means that the astrocytes, as I mentioned earlier, have receptors for neurotransmitters, which with the old concept of glial cells just being supporting cells doesn't make too much sense. But if you just wait for 2 seconds, you will observe that there is a whole communication between these cells, and this is immunofluorescent calcium. The plate is full of immunofluorescent calcium, and you can see this a slow but consistent communication with these cells. So something that I think is very important for us to understand, these cells that I have introduced into this milieu are no neurons, and therefore they are not communicating through electricity. They communicate through hemichannels that transport calcium, and therefore the slow communication that they have compared with the light speed of the neurons, it cannot be recorded by the use of electrons. This is, I don't know if you're familiar, Douglas Field. He has worked extensively on The Electric Brain is the name of one of his books, but he has also worked on The Other Brain, in which he talks about glias and neurons and the whole neurodegenerative field in which schizophrenia, depression, epilepsy, and many other conditions are intricately related to the activity of these glial cells. So our understanding of the central nervous system has changed dramatically over the last 5 decades. And part of that, or part of the problem, is that because the glial cells cannot be recorded normally with electrodes for action potentials, therefore we have ignored the presence of these cells and their role in pain are therefore in neuromodulation. But it is very important to understand what is their role and what is the response to electrical signals, because glial cells do not only sense the action potentials that are going through the neurons, they actually control these electrical signals that the neurons are sending to one each other. This is a study that we did about 4 or 5 years ago, and what we were looking is, if we always put our electrical contacts between certain regions, typically for low back pain between T8 and T11, what is underneath those segments? It seems like a very easy thing to find just doing some reference search. You will find out what is the population of cells in that area. Interestingly, we found that that has never been described. So basically, we have, since 1967, we have been putting electrodes, we have been turning them on, and then we have turned around to the sales rep and asked, hey, can you please stimulate? And then when the patient got paresthesia, we felt that we had done something dramatic to improve our patient's life. But in reality, if you look at what you are stimulating, if you just, before you turn around and try to turn all these circuits into acting, you may want to think, what am I stimulating? We always thought that they were neurons, but neurons, we find out that after using a specific immunofluorescence for different cell types, neurons, microglia, astrocytes, and oligodendrocytes, neurons only represent only 8 to 5% of the cells that are underneath your electrical signal. This is the animal model that I have been using since the early 2000s. And right now, over the last 10 years, we have incorporated more and more molecular techniques in such a way that we can evaluate transcriptomics, proteomics, and phosphoproteomics. That's what we are doing right now. And when we did that, one of the first observations is that according to the gate control theory, the activation of the A-beta fibers will generate a downregulation of synaptic-related processes. That's kind of part of that. And we confirmed that. We observed that if we use in the SNI model, which is a model of pain behavior, we could produce downregulation of ion channels, and we could also produce downregulation of synaptic transmission. But something was quite interesting is that after 48 continuous hours of stimulation, there was an upregulation of inflammatory processes. And I don't know if any of you have done neuromodulation, but something that you will observe is that most of the patients respond in the early phase, but maintaining for long-term this alleviation of pain is quite challenging. And that may be part of the clue. Imagine that you are using the gate control theory and then you produce downregulation of synaptic-related processes in pain, but at the same time, you continuously keep pounding the inflammatory processes in such a way that you are kind of shadowing the benefits of this A-beta fiber activation. This is a study very similar to ours. They use RNA sequences. We use microarray. And they observe exactly the same thing: upregulation of inflammatory processes, downregulation of neuronal-related processes. So what we try to do is, okay, I know what low frequency does. Can I dissect the electrical signal? And by doing that, can I try to determine which genes get activated or deactivated with particular frequencies? The only thing that we are changing here we maintain the same type of balancing. So we produce an active cathode, we produce an active anode in such a way that there is an electrical balance. Otherwise, there will be Faraday reactions and damage of the tissues. So we do the same thing, but the only difference is that the time of recovery of the electrons from the field could be symmetric or asymmetric, or it could be totally cathodic or totally anodic. And what I want to show you here is just by changing this parameter of the waveform, we observed that there was a totally different correlation of expression of different genes engaged in immune function, glial modulation, extracellular matrix, and redox reaction. If you are bored one day and you don't have anything to do, you cannot sleep, I always suggest people to do a quick PubMed search, look for my articles on basic science and read them. They are really good to go to sleep, but also I think they can give you a little bit more of all the different details that we have looked like. One of our latest papers have been focused on the extracellular matrix modulation with different components of the electrical signal. So what we came up after years of research is that there were some signals, some components of the signals, frequency, amplitude, pulse width, or recharge balancing, that generated different types of effects in different types of cells. So we identified like fingerprints for each one of these different cell populations. And when we started combining signals, those signals that produce more reversal of the effects generated by pain, and we put them together in what we call multiplex signals, we were able to generate a transcript heat map that resembles those of the first column. In this case, the first column represents the animal with no pain versus the injury. And here you can see how, for example, low rate, as I explained earlier, was going to reverse those inflammatory processes in the wrong direction, while different signals, the combination of signals or high frequencies, had a much better pathway into returning those genes, these transcripts, towards the naive state. So we did, at the same time that we did all this molecular work, we were also looking at the behavior in animals, and we observed that we could have a much better control of pain, which is kind of interesting. You control the transcripts and reverse them towards a naive state, and kind of the next observation is that translates into improve in the hypersensitivity to mechanical temperature hypersensitivity. And we observed that in rodents, we observed exactly the same type of behavior in the ovine model. So our formulation at that point is, why don't we apply different signals simultaneously, trying to target different cell populations? So beyond the neurons, we try to stimulate the glial cells and try to understand what our electrical signals did to that, so we can have a modulatory effect at the level of the neuroglial interaction, always keeping in mind that the glial cells represent the large majority of the cells in that segment of the spinal cord. And if you ever wonder what happens in other areas of the brain, you may want to read Brazilian scientist Hortelano, who has described the different percentages of cells in different areas. In the cerebellum, for example, there is 3-to-1 neuron to glial ratio. So it depends where are you stimulating. So you need to understand what is the concentration of these cells. And now I finally come to probably what everybody has been working. Okay, we see patients every day. Our goal is to try to treat their pain. So everything you have been telling us, I mean, good for rats, good for the ovines, but how about our patients? So after doing the animal studies, we decided that it was time for us to try to apply this working hypothesis and formulate parameters that could be useful in humans. First, we did a feasibility study in 7 different sites with a total of 25 patients. It was only evaluating the response of these electrical signals in patients with axial low back pain. Let me emphasize the word axial. And we were looking into whether or not it was better than conventional paresthesia-based stimulation. After the trial, feasibility trial, we decided to run a randomized control trial, which was on-label, post-market, multi-center randomized control trial. And the idea, like in most of the studies on spinal cord stimulation, was to do a non-inferiority analysis between 2 active groups. One was the paresthesia-based stimulation, and the other one, the application of multiple signals, nowadays called DTM. And then after the non-inferiority was proven, then we were doing application of superiority analysis to determine whether or not that was useful. We did the study in 12 sites in the United States. 128 patients were included in the analysis, and we look beyond the axial low back pain. We also look at leg pain as a secondary outcome, and other outcomes as the Oswestry Disability Index, quality of life inventories, patient global perceived effect, and patient satisfaction. It is quite interesting how statistics are managed nowadays. How do you manage intention to treat analysis in this type of patients? Unfortunately, many of you are facing it right now. For the residents, will face it relatively soon. Insurance companies very often decide who gets one treatment or another. So if you screen 100 patients, out of those 80 patients qualify, many of those patients will not be able to have the therapy because this is a post-market study. So if you want to do an intention to treat analysis and include all the patients, you will be facing some uncomfortable realities. Maybe 30 patients were not approved for the trial, or even worse, they had a successful trial, but then the insurance didn't want to pay for the whole procedure. So most of the data that I'm going to be presenting in the next few slides relate to modified intention to treat analysis. What do we call that? Patients that at least go into the trial phase. If the patient goes into the trial phase, the patient finishes the trial phase, let's say they don't have a dural puncture or they don't have an infection or something through the trial, then those patients that provide data at the end of the trial, all of them will be included in the analysis. And that's what we did. I already mentioned that we did a non-inferiority, looking for a 10% margin. And then after a successful non-inferiority analysis, then we did a secondary analysis looking at responder rate. Responder rate means that the patient needs to have at least 50% reduction in the visual analog scale compared with the baseline at 3 months in some studies, or in others that I'm going to show you at 6 months. This is the baseline demographics. What I think is interesting is that the mean age is around 60. And I will tell you why I think it's interesting. And the duration of pain is around 13 years for those patients. So that means many of these patients had been in pain for a long time, which people in the past used to believe that duration of pain had to do something with the positive or negative response to this therapy. And they also had, in average, 1.5 surgeries. This is what is nowadays called PSPS2. So I'm going to try to refer to that. It used to have other names, but let's call it PSPS2, those patients that for whatever reason after surgery had not achieved the expected results. So here are the primary endpoint, the 3-month outcome. And we were looking at the difference between the use of combined signals with no paresthesias versus paresthesia-based stimulation. We had 51 response rate with the use of paresthesia, which was higher than any other study published at that point. But our group had 80% responder rate, patients with more than 50% reduction in their pain for the axial low back pain. I'm not talking about radicular components. And this is important because we have a lot of different techniques in pain management, from epidural steroid injections, radiofrequency, and other things that can potentially help for radicular component. But the axial pain, I would say, at least for us, is the main challenge. Interestingly, the results were maintained almost exactly the same at 1 year. Also, when you look in this study at the percentage of patients that got what we call profound response, that means more than 80% reduction in the pain. When you use traditional stimulation, 3 out of 10 of these patients got more than 80%. That is good. But if you look at the combination of signals, we were able to achieve 65%. 7 out of 10 patients were reporting a profound response. This is the reduction in axial low back pain. This is the reduction in leg pain. We got a trend. I hate the word trend. It basically means that you didn't achieve what you wanted, but there was a trend also for lower extremity pain reduction. This is a second study, and there is a reason I brought it. It's a study that we finished around 3 months. It's a prospective randomized controlled trial done in the United States. And the main indication are patients that do not have a surgical indication for the pain that they are suffering. So as a big picture, we exclude patients that had facet spondylosis, patients that had, for example, instability, that's the main one, or severe spinal stenosis with neurogenic claudication or things like that. Once you select these patients, and here are the inclusion-exclusion criteria, then we decided to do the same type of a study that I just mentioned, DTM programming versus conventional stimulation. And at 6 months, we allow these patients to cross over. Keep in mind that the final results of the study will be presented next week at the American Society of Regional Anesthesia. That was the presidential award for this manuscript. And I'm not going to present all the data, but I'm just going to present a little summary of what we observed. We had 51 patients based on the MITT population in the test arm versus 54 in the control arm, and we follow the patients up to 12 months. Same thing, as you can see, the age of the patients is the same, but these patients have not had any back surgery. The years of pain duration are around 9 years, a little bit less than those patients that had previous surgeries. And all of them, 100% of these patients had neuropathic pain. At 3 months, which is the primary outcome, we observed independently of the type of analysis that we did, whether we did per population, so that means only the patients that reach a particular point or patients that are implanted, or MITT, as I described earlier, the group with the combination of signals had a much higher, statistically superior response for axial low back pain than the control group. That also translates in something very interesting, that the study from Europe that I'm going to present in a second, I'm not presenting the data for the EQ5D. In the EQ5D is a tool to evaluate the quality of life of patients. Keep in mind, if you have 0, means you are dead. If you have 1, means you are absolutely fantastic. You have no problems in your life. Americans describe the quality of life with the same amount of disability, with the same amount of pain as a 0.5 versus Europeans, who reported about 0.3, exactly the same patient population. I find it quite interesting. Any improvement of more than 0.1 in this scale means that there is a clinically significant improvement in their quality of life. We were able to observe 0.26 improvement in this scale. And I just mentioned very briefly, this is going to be, this was presented about 8 months ago at the World International Neuromodulation Society meeting. This is the European trial. It's going to be the first multi-center, multinational randomized control trial looking at the effects of a spinal cord stimulation. In this case, once again, we look at patients with no previous back surgery. The control arm here is conventional medical management, which means patients have failed conventional medical management, and as a benefit to be part of the study, you let them be in conventional medical management, but they have the opportunity at the primary outcome, which was 6 months, to cross over. So the primary outcome at 6 months, we observe significant difference. This is a superiority analysis between the two groups, and we also observe 24-point reduction in the Oswestry Disability Index. Why am I presenting that? The only reason I'm presenting that is because within the sites in Europe, 3 sites where the PIs were neurosurgeons. And interestingly for me, the outcomes of those sites are much better than the other sites where anesthesiologists or other type of pain physician were handling the patients. And I think it has to do a lot with the selection of these patients. And I think that hopefully in the future, I know some of you are into neurofunctional specialty, but I hope that more and more of you get into this field, mainly because the original concepts that I discussed about the role of astrocytes had a lot to do with the neurosurgeons or brain studies. So I think that you have the tools to decide, okay, this patient is a surgical candidate, this patient is not, and maybe I can have a big impact in their life, preventing a surgery that you don't think is necessary for your patients. I was going to talk about a big hot new topic, which is the ectopic compound action potentials, is the measuring of action potentials as you stimulate. Basically, you stimulate and you generate action potentials. But because of time, I'm going to leave that out. We are working right now in our lab trying to use these concepts of multi-signals, all of them engage with the observation of the action potentials, which is allowing us to determine what is the right intensity independent of the patient's movements, which can make the lead be closer or farther away from the spine. I would say this is probably a whole other presentation. I'm just presenting you here because I like my rats and I'm very proud of what we are doing with them and how we have been able to somehow correlate this concept of action potentials with the other concepts that I have been explaining for the generation of these multiple signals. There is an ongoing study in Australia right now. The study is getting into the 6 months, but the 3 months results show dramatic improvement for those patients that use the combination of the ECAP measurement with the differential target multiplex. So I'm coming to the summary, and I would say something that is quite obvious. We need to understand what is the condition that we are treating in order to be able to treat that condition. I always say, I have all my life, I have been in the field of medicine, so please don't offer me to try to fix your car. I had no idea what's going on, and therefore the likelihood that your car will run after my repairs are extremely low. So we need to incorporate these neuroinflammatory ideas or concepts that are behind the development of this pain. And that is the only way that we could move forward into the improvement of these programming therapies that we give to our patients. There are more cellular elements that those originally described in the neuronal doctrine, and every single cell in this planet, whether it's eukaryotic or prokaryotic, please keep in mind it has a membrane, and as such, it has a different content of ions in and out of this these membranes, and therefore, if you apply an electrical signal, you will have some type of effect within those cells. We did a study years ago with Leishmania cells in vitro, where we observed that you could modulate molecular biological processes through the application of electrical signals. We have been focused on only measuring action potentials, but these glial cells do not produce action potentials, and therefore we have been deaf to the effects of the electrical signals on these other cell populations. We are in a phase right now where we are trying to integrate all these concepts with new technologies, because that's what they are. The measurement of these ectopic compound action potentials are just new technologies that hopefully will help to improve the therapies that we apply to our patients. So what I propose is to change totally the paradigm that we have been working on for the last 50 years. We should understand what the pain mechanisms are. Once you understand that, the next step in my mind should be to understand what electrical signals do to these pain mechanisms. By doing that, you have to separate the effects of the frequencies, amplitudes, the way you recharge your signal, and the intensity of the signal. Only when we do that, we will be able to provide pain relief to our patients. And once again, it's great for me to somehow be back at the University of Miami, even if I am, I think it was like 16 miles last time I was waking up at 5 a.m. driving from Pembroke Pines to Jackson. But thank you very much for this opportunity.
Ricardo, this was just amazing. I loved your talk, really. And you think you're genius, because yeah, over 90% of the cells we have are glia, and we don't get to study them as much as we should. And because neuromodulation is basically empiric, like you stimulate here and there, you get an effect, and then you try to understand what happened. So looking into the effect of neuromodulation through immunologic eyes, like immunology that you master, it's really I think it's going to bring a revolution for the field, not only for pain, but if we could use the same approach to understand the effects of neuromodulation for movement disorders, epilepsy, and etc. I think we're going to have, you are already revolutionizing. And regarding to your approach with patients, because we have it's important to remind the residents, we have PSPS type 1 and type 2 now. We don't talk more about failed back surgery syndrome. We talk about persistent spinal pain syndrome type 2 when they had surgery, and persistent spinal pain syndrome type 1 when they didn't have surgery, but they do have chronic back pain, for instance. And the fact that, because classically we implant patients that went through surgery and still have pain, but the results are very encouraging to treat patients with neuromodulation that never had surgery. And that was what we were discussing in the beginning before we started the talk, that the participation of neurosurgery in this decision, I think it's key to have a great patient selection. That's the most important point to achieve good outcomes. Because if you don't have a clear understanding of the spine biomechanics to select the patients that have some degree of instability, of neurocompression, or other surgical conditions, you of course you're not going to have great results. So I think this approximation with pain medicine yourself and neurosurgery is going to be a really interesting thing that we should work on.
Thank you, Joachim. Two things that I want to emphasize. I hope this is recorded.
It is.
That's wonderful, because you said that I'm a genius, and I would love to show that wife. Maybe she will start thinking I'm not as dumb as I look every single day. And the other thing that you mentioned is that hopefully through this search, we will look into a resolution. I hope not. That is the antithesis of science, and it will not be fun. I cannot tell you how many dead ends I have found over the last 20 years. And I hope to keep finding dead ends, otherwise science is not fun. That's what I don't think we have optimized, and we hopefully will never optimize. There will always be new things coming up and changing our thoughts about what we are doing. But again, thank you very much. And please give me just the little segment that says I'm a genius. I'm going to record that and give it to my wife.
Ignacio can help us with that. Okay. Thank you once again. Dr. Dietrich is on the call. I see him.
Yeah, good morning, everyone. That was a great talk. If you go over to the Lynn Center, we have about 6 studies going on, primarily in spinal cord injured subjects, including neuropathic pain with neuromodulation. And so it's a very exciting area, and I agree, neuromodulation compared with exercise can modulate the immune response and all kinds of other things that are beneficial for our patients. So hopefully you can tour the Lynn Center in the future, and we can talk about some of the studies that are actually ongoing. And we're utilizing human biomarkers to assess the efficacy of these programs and enhancing hand function. And we just got new funding to enhance walking with neuromodulation as well. So a lot of exciting things. And thank you very much for your very interesting presentation.
Thank you, Dietrich. This is fascinating. Let me tell you that our conversation at the Fontainebleau with Joachir, with a couple of caipirinhas, started with the spine efforts that you have been leading for so many years. And we did a paper on the modulation of microglial cells from, we humans need definition, so we define them as M1 or M2. Meaning that M1 is good, M2 is bad, but nature is much more beautiful and complex. There is a whole spectrum of activation of these cells. And one of the things that I was telling Joachir, again, please forgive me, I had probably 2 caipirinhas by the time I was mentioning that, but it was the potential ability in the acute phase to modulate the expression of more friendly cells versus more M1 type of injury, pro-inflammatory cells. At least in the animal model, we were able to modulate these signals, so you can have a more, let's say, anti-inflammatory signal at the beginning, and then change it and let nature take its course. Unfortunately, nature for spine injury doesn't go in the direction that it should, and basically, in order to control the magnitude of the injury of the spine, it ends up generating more damage. So we were just talking, okay, will it be possible to apply very early the electrical signals instead of doing later on in the process? And there is, again, I don't understand exactly what will be the final effects, but there is definitely a potential value of trying to modulate early on before the whole process of neuroinflammation happens in the injury, to avoid some of the later, more difficult to treat consequences of the injury. So.
Yes, we agree 100%, and we're actually now using exosomes derived from human cells that actually affect the immune system, and you can actually infuse these, multiple times using IV approaches. And they seem to modulate the angry microglia, for example, after traumatic brain injury and spinal cord injury. So we're doing ways that we think at the bedside can make a difference in the very early inflammatory responses to stress and injury and things like that. So a lot going on, a lot of opportunities for collaborations and things like that. So thank you very much again for a very exciting and interesting talk, and we should continue these discussions at another time. Now, we've got another exciting presentation coming up. Dr. Nadine Kerr. Nadine is one of our more recent recruits. She's a research assistant professor in the Department of Neurological Surgery, and a very important component of our MAMI Project mission. Now, recently, Nadine is rewriting the textbooks in terms of how focal brain injury or spinal cord injury can affect multiple organ systems. We now know our patients after stroke or traumatic brain injury and spinal cord injury over time have multiple organ problems, and we're trying to understand, in addition to neurogenic mechanisms, what other mechanisms could be involved in these remote organ system changes that we see in the lungs, we see in the gut, and the heart, and other organ systems. So Nadine is going to talk today about some of the work she's been doing in stroke and Alzheimer's disease using preclinical models. And it's really an exciting area that opens up new strategies for trying to modify these biological processes that are occurring in our patients and improve overall outcome. With that, Nadine, thanks for joining us today and look forward to your presentation.
Okay, well, today I'm looking forward to talking to you all about some of our recent work on the gut-brain axis and how it is affected after brain injury, particularly stroke. And more recently, we have started to examine as well the relationship between TBI and stroke and Alzheimer's disease. So as I'm sure many of you know, stroke is one of the leading causes of death worldwide. And just to share some recent statistics that came out in 2021, 1 in 6 deaths from cardiovascular disease was due to stroke. And every 3 minutes and 14 seconds, someone dies from stroke in the US. 87% of strokes are ischemic, which is the model that I'm going to be discussing with you that we study in the lab. Stroke-related costs in the United States came to nearly $56.5 billion between 2018 and 2019. And stroke, as we know, the risk increases with age, which is important for our studies in Alzheimer's disease. And stroke also reduces mobility in more than half of stroke survivors that are 65 and older. So to bring in Alzheimer's disease, it's the 7th leading cause of death in the United States. More than 6 million Americans are living with AD. 1 in 3 seniors dies of AD or dementia. And there's a lot of talk about genetic predisposition to AD and early onset. And so the risk for AD is at age 45 is in 1 in 5 for women and 1 in 10 for men. So as we also know with stroke, it is more common in older females as well. So this is also going to be important for some of the data I'm going to show you. About 1 in 9 people that are age 65 and older have AD. And the deaths from Alzheimer's have doubled between 2000 and 2019. So making it a really important area of research. So the relationship between CNS injury and AD has become more, there's more research in the past several years. We just had a graduate student that recently defended who studied TBI, particularly the genetic predisposition to TBI and how that progresses the onset of Alzheimer's disease and AD pathogenesis. So seniors that have had a history of moderate TBI have a 2.3 times greater risk of developing Alzheimer's. And those with a history of severe TBI had a 4.5 times greater risk. So showing that there's an important relationship between the two. As well, adults with traumatic spinal cord injury are at higher risk for AD, and more importantly, for background that I want to give you for this talk, is that stroke, both ischemic and hemorrhagic, are risk factors for developing AD. And about 10% of patients who suffer from an attack of stroke for the first time have dementia. So as Dr. Dietrich was mentioning, the focus of my work and the past several years has been in traumatic brain injury, severe brain injury, and systemic complications. So I started out looking at this, focusing on severe TBI and the very acute time points after severe TBI, particularly looking at lung injury, acute lung injury, and acute respiratory distress syndrome. And as continued then to study systemic complications after stroke, and after looking at a lot of clinical data and talking to a lot of our collaborators in the gut world that study gut research, there are one of the major systemic complications that occurs after stroke is in the digestive system. Now, after stroke and any severe brain injury, of course, there's acute systemic complications, and in stroke, same as TBI, a lot of those are seen in the respiratory system, including lung injury and pneumonia, as well as in the cardiovascular system. In the digestive system, of course, there are some acute complications that can occur with stroke, but more we see these as chronic complications and disorders that develop over time. And there's actually with systemic complications after Alzheimer's disease, I was realizing while getting ready for this talk that there is a lot less out there about systemic complications and Alzheimer's disease. One of the reasons might be due to the lack of reporting And also, as opposed to a CNS injury, there's no actual injury that occurs, so that these acute phases that we can study are not as obvious in Alzheimer's patients. But what we do see is that a lot of these systemic complications are chronic and progress slowly, as does the disease itself. Some of the chronic, since I was talking about the lungs, one of the more common chronic complications that occurs is obstructive sleep apnea and COPD. And then as well, there's this systemic inflammation, which is really important to our area of research, that occurs and starts seeing in the circulation. And then this can affect multiple organs. Again, what we're very interested in is in the gut. And in particular, with Alzheimer's patients, the focus on gut research has really been on the microbiome and gut dysbiosis. And while, of course, that is a very important component, we're really interested in seeing how the systemic inflammation and the inflammatory response in the gut causes gut disorders in Alzheimer's patients. So I thought this was interesting because as I was reading and almost kind of went down a deep dive on the history of the gut-brain axis, because in recent years it has become very popular, but for a long time, and whenever I talk about this, I like bringing up that we've for so long been saying we have a gut feeling. So as I did some research on the history, I was kind of surprised that the first time any gut research was done looking at bacteria in secretions, so in both stool and saliva, was in 1681. And that was the first time a bacteria per se was discovered. Then the research on this was left alone for almost 2 centuries. And in the 19th century, people started talking about, scientists started calling the gut, there was a nervous sympathy. And this was sort of the first time that the idea of the gut-brain axis came up. And there was general public sort of looked at this like it was not true. And a lot of scientists were saying the way they described it was that the negative feelings, our negative emotions were in the gut, while our positive emotions were in the heart. So there was that sort of the start of the brain and other organs. And but again, like in this picture here, was a lot of satire about this whole idea. And then in the 20th century, this was the first time by this French scientist that probiotics were developed. And again, he was sort of, looked down upon because he was saying that taking probiotics would maybe help mental health. Now, as we move on to the present, we see that in the past, since the 21st century, there has been a major increase in the number of papers that have been written on the gut-brain axis. There's this little area here where they went down, but I think that was due to COVID. But really recently, the first areas where it was studied was in psychiatric disorders. So just some of them are depression and anxiety, autism, and schizophrenia. And then it moved into neurodegenerative diseases such as AD, Parkinson's, and multiple sclerosis. And more recently, which is the focus of a lot of our new work here, has been in SCI, TBI, and stroke. So some of the gut-brain axis disorders that we see in stroke, there's actually 50% of stroke patients experience some form of GI complications. The most common, gut dysbiosis, so changes in the gut microbiome, leaky gut, which is can be described from various disorders and is basically changes in the gut permeability, thus allowing toxins and to come inside the gut. As well as in some of the more acute complications is gut hemorrhaging and gut sepsis. And then another symptom that a lot of stroke patients see is chronic constipation, fecal incontinence, and then the 2 most common gut disorders seen chronically in stroke survivors is irritable bowel syndrome and inflammatory bowel disease. So as I mentioned before, in Alzheimer's, there's been less known about the specific disorders that Alzheimer's patients suffer from, but research has shown that chronic GI problems, such as irritable bowel syndrome and inflammatory bowel disease, as well as gut dysbiosis, those patients that with Alzheimer's disease that have these GI complications actually have an increased risk of dementia-related disorders. So that sort of gets us into the bidirectional aspect of the gut-brain axis, as you can see here. And so on this side, we just see what a healthy gut-brain axis would look like. So normal gut microbiota, normal intestinal epithelial barrier, as well as blood-brain barrier. So we have normal healthy levels of inflammatory cells and mediators, and the composition of the microbiota is normal, and as well as cognition is as normal as well. However, getting into Alzheimer's disease, there's intestinal dysbiosis, the blood-brain barrier, as well as the intestinal epithelial barrier are impaired. And we start seeing in the gut, as well as in the brain, changes in pro-inflammatory mediators, as well as microglial activation, which is an area that we've been studying for quite a while in our lab, and I will talk about in a little bit. So all of these issues can cause dysregulation of homeostasis, leading to changes in the gut microbiome. And then we also see, as we know with Alzheimer's patients, alterations in behavior and cognition. Okay, so as I just mentioned, the bidirectionality of the gut-brain axis is really, really of interest to us. It also, as there is more coming out about the bidirectionality between other organ systems and the brain, and it does make it interesting yet somewhat difficult to study to understand how exactly these changes, that the signaling is happening between the brain and other organs. And that is something that I will discuss. And another important thing is that as with all systemic organ complications and the communication between the brain and other organs, there are several pathways that will contribute to this. And that is something that we acknowledge. And of course, our focus is the immune system. However, it's important to note and keep in mind that there is changes, and the role of the neuroendocrine system is important, as well as the enteric nervous system, the circulatory system. So especially when we see inflammatory mediators in the circulation, and the role of the vagus nerve. And I also highlighted this because the secretions, I'm going to talk to you a bit about exosomes and extracellular vesicles. However, all of these secretions, several of them can actually be composed in exosomes. And so a lot of these secretions that are secreted after stroke or in Alzheimer's disease from the brain and then traveling to the gut, those are very important for communication. So in order to study the systemic inflammatory response, we first sort of just want to go over the inflammatory response in the brain after CNS injury. So immediately after injury, within the first hours and minutes post-injury, there's a release of alarmins. So some examples of alarmins are damage-associated molecular patterns, such as HMGB1, IL-33, ATP, mitochondrial DNA. And once these alarmins are released, they will activate the innate immune system, which is our main area of focus, particularly the inflammasome. The inflammasome is an important component of the innate immune response, and I'll get into that in just a second. And so once the inflammasome is activated by alarmins, then we see the release of cytokines IL-1, such as IL-1 beta and IL-18, amongst many others. And then within the first minutes to hours post-injury, neutrophils arrive and can often last 2 days past post-injury, as well as we start seeing the infiltration of monocytes and lymphocytes, which can then stay for days to weeks post-injury. So the inflammasome, as I just mentioned, is an important component of the innate immune system, and it is a multi-protein complex. You can see it here, and these are the different inflammasome sensors. So there's the main one that's really there's a lot of research on is NLRP3, which we study quite a bit, as well as AIM2, which we previously studied in traumatic brain injury and pulmonary dysfunction after brain injury. So also seems to be an important one in systemic complications after brain injury. So once the inflammasome sensors are activated, then these bind to the to caspase-1 and through an adapter protein, which is apoptosis speck-like protein containing a caspase recruiting domain, which is, we call it ASC, and what I will continue to call it. And then ASC, after the binding and the creation of this multi-protein complex, we see the activation of caspase-1. And then caspase-1, the active form of caspase-1, will then cleave gasdermin D, which is involved in pore formation in the cell membrane, known as the gasdermin pore. And then this leads to a form of cell death known as pyroptosis. And that then leads to the cleavage of IL-1 beta and IL-18. And after the pore formation and cell death, these cytokines are released into the extracellular space. So just to quickly compare pyroptosis to other forms of cell death, we know that apoptosis involves a series of tightly controlled events, which is characterized by cell shrinkage and membrane blebbing and changes in organelles and organelle loss. Necrosis, which is stimulated by a number of external factors, such as infections and toxins, and that leads to morphological alterations and ultimately cell death. And then pyroptosis, which you can see here, the pore formation that I was just mentioning, this is activated through the inflammasome, and this leads to a rupture of the cell membrane, which then releases the intracellular contents. So to study all of this and the role of the inflammasome and pyroptosis in our lab, we use a photothrombotic stroke model. So this is this model is done using a laser, and it aims to induce ischemic damage within a given cortical area. And this is done by means of photoactivation. And we do this by the injection of a photosensitive dye called rose bengal. We inject it into the mouse via the tail vein. And then once we irradiate, then the dye is activated and it produces singlet oxygen that damages the components of the endothelial cells and the endothelial cell membrane, leading to platelet aggregation and thrombi formation that will then eventually stop blood flow. So this model was developed here by Dr. Dietrich and Dr. Watson. You can see here this SCM image. You can actually see the platelet aggregation. So we're very fortunate. It was developed originally in a rat model, and more recently, over the past couple of years, we've optimized this into a mouse model. And it's a good model to use. It's minimally invasive. There's no craniotomy, so on an intact skull. We've found that it's highly reproducible, and we have the well-defined localization of the ischemic region for our stroke. And we've also shown, we published a paper a couple of years ago in young mice, which is different here. In this study, we'll be using aged mice, and these were wild-type mice that did not have any signs of Alzheimer's disease. And we did see, what we published, we did see gut dysfunction in these mice. And we've also actually examined some of the other organs. So we do know that this model does produce systemic complications. And the mouse model that we use is a transgenic mouse model. These mice are the 3XTG mice. It's a commonly used Alzheimer's disease model. There's several, but this one is is good because it contains all 3 mutations associated with familial AD. And we've actually recently looked at this in traumatic brain injury and published a paper just a few months ago showing that with this mouse model, these mice, they, as they're genetically susceptible to Alzheimer's disease, if they receive a moderate TBI before the onset of the disease where we see the pathology, the progression of AD pathology occurs faster than our wild type mice. So it's a great model to study amyloid beta, which is what I'll also be showing you some work on that soon. And it's so at 6 months of age is when the amyloid beta AD pathology really becomes apparent in these mice. So as in our previous study, looking at them earlier on is, as they're genetically susceptible, is important to study then future outcomes once they have had the onset of Alzheimer's. For the purpose of this study, we are really interested in the aging effect. So we've used aged male and female mice that are 12 to 18 months of age, which translates to about 55 to 65 years of age in humans. So getting into some of our data, this is what our lesion looks like after the stroke. And as you can see in our wild type mice, this PTS is our photothrombotic stroke. So in our wild type photothrombotic stroke mice, you see very nice infarct here. However, there's a very obvious increase in the size of the infarct in our 3XTG mice. You can also see that it's larger as well. We are still working on getting actual lesion volume data, and we hope to be able to show that soon and publish that soon. And I also thought it was really interesting to show, because working with aged mice in general is difficult due to mortality, as well as we breed these mice here, so colonies, etc. It makes it difficult. But we also, with adding the Alzheimer's component, just makes the mortality factor much more difficult. And as you can see here, there's a higher mortality rate in our Alzheimer's mice compared to our wild type. And as we see in the human population, and we know as both stroke and Alzheimer's disease are more common in aged females, so sort of separated this out by looking at differences in male and female. And there's very obviously, we had a larger number of deaths in our female mice, Alzheimer's mice, as opposed to our wild type, and also in our male mice as well compared to our wild type. And you can even see just with aging alone, so without the component of Alzheimer's disease, our female mice also, there was more mortality in our wild type female mice compared to our male. So as we know, there's of course, to actually validate the stroke model, it's important for us, of course, to do cognitive and motor function behavior. And as we've shown that, again, we published that a couple of years ago to validate our model in younger mice. However, we've also performed some tests in our aged mice. But first, I thought just to see some of the statistics and data on cognitive decline and stroke. We also, as we see that stroke, as I mentioned, increases the risk for AD. And recently, a few years ago, the NIH has really put out a lot more interest in funding and just to have research on the relationship between cardiovascular disease and cerebrovascular disease and Alzheimer's disease and Alzheimer's related disorders. So here, this VCID is known as vascular contributions to cognitive impairment and dementia. And as you can see, we see that there's this overlap between stroke and Alzheimer's disease and dementia-related disorders. So there is a huge interest in this area and the mechanisms of this, because we do know that stroke doubles the risk of vascular dementia. 30% of stroke patients will go to develop cognitive dysfunction within 3 years after stroke. We also know that memory loss is associated with stroke and, of course, Alzheimer's disease. And while there are several risk factors that are for both stroke and Alzheimer's disease, one of the common ones, the most common one, is aging. And in our preclinical model here, our stroke model, we actually see this. We can show this, showing it is similar to what we're seeing in the human population. And this is using 2 of our behavior tests that we perform in our lab. And the first is the open field test. This one tests anxiety, locomotor activity, and exploration. So basically, we have the mice we put them in a large cage, and we examine, as we video them, we examine their exploratory behavior. So what we're measuring is how much total distance they're moved, how much time they spend in the border, which would be more indicative of anxiety and less exploration, and then how much time they're spending in the center. So, of course, the stroke in general, they have both Alzheimer's and AD. They do spend more time in the border and less time in the center. However, we also noticed that the Alzheimer's mice, their total distance moved was less compared to the wild type mice, which could be both really due to motor dysfunction. I don't have that here, but we have shown that with using a rotarod test, we also see decreased motor function in the Alzheimer's mice after stroke. And then we also perform a novel object recognition test, which examines spatial memory, exploration as well, and recognition memory. So where this is a 3-day test where mice are exposed to 2 similar objects, and then the next day we introduce a novel object. So the mice that are, we calculate this based on their how they're examining the new object as well as their exploration. And again, you can see that the wild type mice, both the stroke mice also had changes in memory and decreased memory as well as exploration. And we see that also decreased in our Alzheimer's mice. Okay, so now getting into the inflammasome and the acute inflammatory response. Here we studied inflammasome expression in the cortical tissue of aged and wild type mice, but we see and this was in the acute time point at 24 hours after stroke. We are in the process of increasing these time points and looking out to up to 7 days, which is really where we see where we start seeing that inflammasome expression starts decreasing. But our previous data with the lung injury model, as well as our TBI and AD model, we see that 24 hours seems to be a time point where we notice the most robust changes in inflammasome expression. And as you can see here, so some of the important inflammasome proteins that I mentioned, caspase-1 and cytokine IL-1 beta, are significantly increased in our AD stroke mice, as well as the adaptor protein ASC, and the gasdermin D, which is the protein that is responsible for pyroptosis. So in our lab, we've studied the role of microglia in severe brain injury for years. In 2018, one of our grad students published a paper looking at a penetrating brain injury model and inflammasome expression in microglia. And recently in our TBI-AD work, we've looked into inflammasome expression in microglia and changes in microglial morphology. And microglia have been studied in Alzheimer's disease and are known to be one of the most important cell types, of course, because they are the inflammatory cells in the brain. And here, this chart here shows you how the early protective microglial response is. So here we have the progression of Alzheimer's disease, and on the y-axis, the magnitude of the clinical and pathological abnormalities, which are based on amyloid beta and accumulation, as well as tau tangles. And we can see that when there's a defective in a response or inappropriate microglial response, what they call it here, we see that there's the progression of Alzheimer's disease and the magnitude of the clinical and pathological abnormalities are higher. And this happens with changes in microglial phenotypes. So as we know, when as in the activated and amoeboid state, we're going to have more, the changes in that morphology also lead to increased inflammation. And here, this is from our mouse model, we can see that in the Alzheimer's stroke mice, so the microglia, as you can see here in there's an increase in the activated microglia, both in the wild type mice and the stroke mice, but there's also more co-localization. In the red is for the inflammasome protein ASC, and in the green is the IBA1 staining for microglia. And we see more co-localization of ASC in our activated and amoeboid microglia. And they're also seen in the sham, but there's a clear increase in the number of activated microglia. And we're actually also in the process of doing a cell counting on the different changes in phenotypes and in between our AD mice and our sham mice. Okay, so now that we've looked in these inflammatory proteins in the brain, the main question is, how are these proteins traveling from the brain to the gut and causing gut dysfunction? And our main area, that signaling mechanism that we're studying, is extracellular vesicles, and which the most common one is exosomes, which of course can also be used as Dr. Dietrich mentioned just now as therapeutically. But for the case of this, we're looking at the biological change in exosomes. So extracellular vesicles are cell-derived vesicles. They're enclosed by a lipid bilayer. They're important in cell-to-cell communication as they're released from cells, and they contain proteins, lipids, and nucleic acids. And they're traveling in our circulation. And more, started out, people studied exosomes and extracellular vesicles in serum and plasma. It's been found in CSF. For the purpose of this talk, where the stool is important to us. But really, they can be isolated from any bodily fluid, which also makes them important as biomarkers. Okay, so here's a sort of schematic of our hypothesis for all of our work. So after Alzheimer's disease and stroke, we see inflammation in cells such as microglia, and then pyroptosis and then these inflammatory mediators are then released due to changes in the blood-brain barrier. And we believe that they're in extracellular vesicles, or they're contained in extracellular vesicles, particularly for our interest, the inflammasome proteins and gasdermin D. And we're studying those that are isolated from the serum and plasma. And then these exosomes are taken up by gut cells. And after stroke and Alzheimer's disease, as it is, there's changes to the intestinal barrier. So the intestinal barrier is already more breached and there's more sensitive. So there's more uptake into the gut, and particularly into the gut epithelial cells, where we then see inflammasome activation and pyroptosis in the gut cells. Then in turn, those release inflammasome proteins. This also leads to gut dysfunction. And the way that we can really sort of say that these are derived from the gut is that we're examining the extracellular vesicles from the stool. Because a big question we always get when studying organ-to-organ communication is, how do we know, for example, in the serum, that these are actually coming from that specific organ? So in order to do that, we've in our lab, it's not been stool-derived EVs is not commonly studied, so it took us a little time, but we've optimized a method to isolate vesicles from the stool. And then these vesicles are then taken up into the brain as causing this bidirectionality. And then that leads to further neurological dysfunction. And this would really support the clinical data that patients that have gut complications do also have, after stroke, do develop worsened cognitive function chronically. And we have shown in 2018, we published this paper, and this was taken from patient samples, serum from patient samples, where we isolated the vesicles from the serum, and we also looked at the whole serum, and we found that ASC was increased. And when we did some biomarker analysis, we also found that so ASC was could be used as a biomarker for stroke, but also for importantly for us was that we could really show that these inflammasome proteins are contained inside extracellular vesicles in the serum after stroke. Okay, so the gut dysfunction and one of the tests that we study in the mice is changes to the gut permeability. So what does that really mean? Well, there's a syndrome that is known as leaky gut, which I'm sure many of you had heard from, but it's really what it is changes to the gut permeability. And that occurs by alterations to the tight junctions in the intestinal barrier. And this is really seen in irritable bowel syndrome and IBD, which I mentioned are the most common chronic gut disorders after both stroke and Alzheimer's disease. So the microvilli, as we see here, in a healthy gut microvilli, we see that there's a strong immune system. The barrier with the gut epithelial cells is intact. And so there is a stronger immune system. The digestion is normal, allowing not the bad inflammatory proteins and nutrients to enter the gut. And when there's damage, this is an example of a damaged leaky gut, the microvilli in the damaged leaky gut, where we see here more inflammation, and this is due to changes in permeability of the gut epithelium due to breakdown of the mucus coating, which is one of the important roles of the microvilli. And this allows bacteria, toxins, pro-inflammatory mediators to enter the gut, and that leading to damaged microvilli. So in our stroke model with our Alzheimer's disease mice, we did some histological looked at the histology. And so similar to what I just showed you, in our wild type sham mice, the microvilli look healthy here, which is a sort of a finger-like shape. And we don't see really the presence of much inflammatory cells in the epithelium. But in just our stroke mice, the wild type stroke mice, and these are also aged mice, and we took these samples, we looked at these at 1 month after stroke. So to sort of mimic the chronic changes that I've been talking about. So even in the wild type stroke mice, there is changes to the gut microvilli, and we see sort of a change in the wave-like shape and sort of breaking of the microvilli. And that is also, interestingly, in our sham, even though these wild type age mice had stroke, in our sham 3XTG mice, we see really also a change in the morphology of the microvilli. However, in our stroke Alzheimer's mice, there's a real dramatic change, and the microvilli, we see that they really don't compare to this wild type healthy shape. We don't see that here. And we do see a real increase in inflammatory cells in the gut epithelium. So we also, aside from looking at the inflammation and the inflammasome expression in the brain at 24 hours, we also looked at these inflammasome proteins in the gut. Particularly, this is in the small intestine at 1 day after stroke, so acutely. And we see, again, an increase in caspase-1, IL-1 beta, ASC, and gasdermin D. And functionally, we do an assay to look at gut permeability. This is called a FITC-dextran assay, because normally in a healthy gut, FITC-dextran, due to its molecular weight, is not permeable. The gut, it will not enter the gutter. It's less permeable. And due to changes in the intestinal barrier and the increase in gut permeability, if there is a increase in gut permeability, we're more likely to see the FITC-dextran has entered the gut, and this is done it's a fluorescent assay. So we see that there's an increase after stroke in our wild type mice, but as well as a significant increase of permeability in the Alzheimer's mice. And again, this was also taken. This was due actually at 3 days. At 3 days post-stroke, we're hoping to then examine this at more chronic time points as well. Okay, so moving on to some of the Alzheimer's pathology in our model. So as this 3xTG model, as I mentioned, is really good for examining amyloid beta. And A beta is this, with Alzheimer's disease, there's known as the A beta cascade, and we know it's a critical initiator of AD. The formation of A beta plaques is commonly used, well, a biomarker for AD. It's been found in both plasma and CSF as a diagnostic biomarker tool. And recently, a very recent study just came out that found that there was A beta in the gut of aged transgenic AD mice. So with all of this, what gut cells are actually the ones that are sensitive to changes in the epithelium after stroke and Alzheimer's disease, and are the ones that are responsible for possibly for this A beta accumulation as well as inflammatory changes. So here's just the gut epithelium. There's many cells in the gut. So just to look at the ones that are in the epithelial cells, they all have similar but also different functions. And we have epithelial cells, goblet cells, enteroendocrine cells, and Paneth cells. But the ones that we've sort of started focusing on, and we hope to do some deeper sequencing to look at the other cells as well. But right now we've started focusing on the crypt cells. One of the main reasons because it's been known to have inflammasome activation in gut disorders such as ulcerative colitis, but they haven't been studied really in the inflammatory response in Alzheimer's disease or stroke. So they're important for providing stem cells for the renewal of the intestinal epithelium. So really without the functional crypt cells, there'll be barrier restoration after injury, or in Alzheimer's disease will lead to worsened permeability, and it'll be more difficult to restore function of the gut epithelium. So this is some immuno staining that we performed on our gut sections of Alzheimer's mice and wild type mice 1 month after stroke. And we found that, so in the green, we are staining for EPCAM, which is a marker for a stain for the gut epithelial cells. And in red is amyloid beta. So we found that there is presence of amyloid beta in the gut epithelial cells in our Alzheimer's mice without a stroke, as well as our Alzheimer's mice that did receive a stroke. But what I thought was really, really interesting here was that in our wild type mice, so in our wild type sham mice, we see some accumulation of A beta, which could be just due to aging. But in our stroke mice, we see that in the gut epithelium, there's increase in amyloid beta expression. So that basically saying that after stroke in aged mice that are not genetically susceptible for Alzheimer's disease, that we can see a change in increased or the starting amyloid beta accumulation, and this is in the gut. And this has been shown in the brain in both stroke and traumatic brain injury, but here we're actually seeing this accumulating in the gut. And another interesting, looking back at the inflammation side of this, we've started to grow intestinal organoids and in culture. And these, this is so we can further look at the cell-specific changes in the epithelium, as well as ideally what we're trying to get to is using these organoid model to look at cell-to-cell communication and changes in barrier function and in tight junctions. So this is how we culture the organoids. This is very preliminary, just started this, and we're right now just looking at these in wild type and Alzheimer's organoids from mice and from aged mice. And after we isolate the organoids and we start the culture as they grow with various growth factors, we can see that within almost 8 days, we start to see the presence of the intestinal organoid. And doing some immunostaining here, we found, so in The red is the inflammasome protein ASC, and in the green is the crypt cell marker. So this is LGR5. It's particularly just for crypt cells. And we can see the expression of an increased expression, colocalization here of ASC in the crypt cells in our AD mice compared to our wild type mice organoids. So the last little part I'm going to show you is some work we've been doing with some of our extracellular vesicle work that we've been doing and looking at the components of extracellular vesicles, how they're involved in this bidirectional communication, and potentially it's also important of how we can use these as a biomarker. So here's a timeline of revisions for the protocols of AD diagnosis. This was came out, the most recent revision was in 2018. This is from the National Institute of Aging. And so this first was developed in 1984, where you can see that already A-beta plaques and neurofibrillary tangles were used as biomarkers. However, at that time, it was really the only methods were biopsy and autopsy. And also at that time, it was really just one stage of disease, which we called dementia. So you really couldn't distinguish AD from other forms of dementia. Then in 2011, in the first revision, there's the 3 stages of disease that are, the preclinical, mild cognitive impairment, and Alzheimer's disease. However, the biomarkers, again, the methods were still with biopsy and autopsy. And moving into the most recent revision in 2018, our biomarkers here is the ATN system, as they say, A-beta levels, tau levels, and the neurodegeneration. And this, what we've also now, they're allowed to look, or that's accepted for diagnosis, to not only look at with autopsy, but also and biopsy, but also as the presence of these biomarkers in fluid, which is really good for diagnosis as well as with imaging. Okay, so again, just preliminary data that we're generating. So here, these are, as I mentioned, we have, after some time, figured out how to derive extracellular vesicles from the stool, and this is a nanocyte analysis, which is because extracellular vesicles are very small, 40 to 1,000 nanometers. So they're difficult to see with just immunostaining. And the nanocyte allows us to measure the concentration of the vesicles, and it also looks at the size. So that's really the gold standard to confirm that we have extracellular vesicles. We also looked at various membrane markers for EVs that we did find as well in these vesicles. So we really could say that they are actually extracellular vesicles that we've isolated. And so in the stool-derived EVs, we've seen that they're in our sham as well as in our AD mice, there's an increase in the cytokine IL-1 beta, as well as the presence of amyloid beta. So that's actually saying that we see amyloid beta. As I mentioned, we see it in the gut, but now we're also able to say that we're finding amyloid beta expression in stool-derived vesicles. And then to really tie everything together with the bidirectional axis of this, we have we took the isolated EVs from the stool, and we'd previously shown both in our traumatic brain injury model as well as our previous study in stroke and gut dysfunction in younger wild type mice, that when we isolated the vesicles from serum, we injected the EVs from a stroke mice into a naive mouse, we found an increase in inflammasome expression. So that was suggesting that the EVs are playing a role in the bidirectionality of the gut-brain axis after stroke. And but the second question, which is often asked, is how. So we've seen the top-down approach from the brain to the gut. Now, how are these vesicles then traveling back up to the brain, causing the bidirectional axis? So we isolated the EVs from the stool so that we could more safely say that they're gut derived. And we injected the vesicles from the stool from the injured AD and wild type mice into a naive AD and wild type mouse that has no stroke. And after we inject these intravenously through the tail vein, and we see that after this adoptive transfer, we see that there's an increase in both the inflammasome protein caspase-1 as well as amyloid beta. So again, showing that both inflammasome proteins and AD-associated proteins such as amyloid beta are involved in these bidirectional axis, saying, suggesting that they're contained in inflammasome in extracellular vesicles, and that this is a potential signaling mechanism for the changes to the bidirectional gut-brain axis after stroke and Alzheimer's disease. So currently, treatment in general for gut-brain axis disorders, there's it's a combination of things such as lifestyle changes, exercise, dietary approaches, of course, because the microbiome is very important. There's probiotics and prebiotics that are being used in both CNS injury patients that have gut complications, as well as Alzheimer's disease patients and in other diseases such as Parkinson's disease. And again, cognitive behavioral therapy, since this is bidirectional, that is also used. But we need to include and look at this as a more as an approach that's looking at all of these components that may be targeting the inflammatory components, such as the inflammasome, the enteric nervous system, which we hope to get into eventually studying, and again, maybe uptake or inhibiting uptake of extracellular vesicles, or just targeting the cell signaling mechanism of these extracellular vesicles. And some of the immediate sort of future directions that we're going to take with this study is we want to further examine the content of the stool-derived EVs, and for really interested in studying their use as biomarkers for stroke and Alzheimer's disease, looking at differences between the young and aged mice, as well as the female and male mice. We're going to continue growing our organoid model, and then looking at some therapeutic intervention options, such as some inflammasome protein inhibitors, as well as some inhibitors that target the formation of pyroptosis of the pore membrane, leading to pyroptosis and extracellular vesicle uptake. So with that, I just have to acknowledge all in our lab, as well as Dr. Bramlin and Dr. Dietrich, who I've worked very closely on this for several years, Dr. Watson for helping us with the stroke model, and our collaborators with the inflammasome group, Dr. Keenan, Dr. Vakari, and we've worked with some really big experts in the gut field. Dr. Donner and Dr. Subita Roy has recently been helping us with our intestinal organoid model. And that's it. So thank you for having me and listening.
Thank you, Nadine, for an outstanding presentation. It's really interesting, right? How the brain and the central nervous system interacts with all these other organ systems in a pathological state, and how it can affect, your overall wellness. So I think it's a really interesting area. So we look forward to your continued studies. Any other questions or comments from the group, please? And we congratulate again Nadine on this new Alzheimer's grant, and she's waiting to hear back from NIH on her first R01 submission. So wish her best of luck in that as well. So clinical translation, what do you think? How you approach this in terms of individuals living with stroke and AD?
I think, I think looking again, right, I know right now we're really looking at the inflammatory response, but I think it's important, and for such a big undertaking and studying the gut-brain axis, to really collaborate with other people on the enteric nervous system. I know in the past couple of weeks, we've heard a few people talk about how important that is, and then incorporating some of these mechanistic things that we're finding with approaches that we can already use, such as diet and lifestyle changes. And I think targeting the gut earlier on after CNS injury will be beneficial to some of these chronic changes that we're seeing, especially with our patients that already have Alzheimer's disease or mild cognitive impairment that could progress to Alzheimer's disease.
Very good. Okay, I see no comments or questions. So Nadine, thank you very much. And everyone have a great day. Take care. Bye-bye.
Thank you. Bye.